Hi guys, Bill here again. Today I'm going to talk about PCR. We're going to talk about what PCR does and what PCR um, is, and hopefully you guys can understand more about PCR. We can look at this diagram, and this describes what polymerase chain reaction, which PCR stands for. And hopefully, looking at this diagram, you guys can recognize a couple things going on, but I don't expect you guys to know details. And hopefully, learning about the steps and how the PCR works, hopefully, you guys can have better understandings of how this steps of PCR works. Let's talk about first what PCR is. The definition of PCR stands for a technology which allows us to amplify single or multiple copies of DNA pieces to make millions of copies of DNA sequence. In other words, we have this single or two or a couple DNA samples, right? When we want to make bunch, you know, um, like for example, like you need couple of couple of bags of sugar, but you only have one sugar. You have to go to a store to buy four more bags of sugar to cook this cookies. But you can't do that without four other bags of sugars, right? So we need to amplify for same thing for genes or for DNA. We might need for certain cases, we might need more DNA, so we use PCR to amplify it. Purpose. So what is purpose of PCR? We kind of got the gist of that. We start thinking about that when we think about the definition of PCR. But what exactly is PCR then? Oops, go back. So I'm gonna let you guys give about 3 seconds to think about that. So if you need more than 3 seconds, please pause the video and think about what might PCR be used for. Okay, so the purpose of PCR is can be a lot actually of many things. It can be found in a bunch of applicants in science actually, but on main things can be found in medical and biological lab uses. So, like the example I talked about back here, talking about that definition. So you might need 4 or 5 bags of sugar, but you only have one to make cookies. So you might need buy more, right? Same thing as DNA. You might need um, thousands, millions copies of DNA to help analyze certain genetic disorders or to sequence certain genetic information. We can't do that without most oftentimes thousands and millions of copies of DNA. So we use PCR. Another example I can think of is forensic science. Um, we often find you guys probably watch crime TV shows often and hear about DNA testing. And there's leftover trace of fingerprints or blood, but those fingerprints and blood might not have enough genetic information, DNA. So we have to amplify it. We have to make more so that those scientists can um, genetic te be able to genetically test those fingerprints for genetic fingerprints or even paternity testing. So this is where the PCR comes in. So what do we need to start the PCR? So there's many things we need, many components you need for PCR, and this is really important because this is key components for PCR, and I believe that every classes that I've taken usually makes you memorize these components. So I highly suggest you guys look into these components because without these, you can't really start PCR, right? So you need DNA template, two primers, tech polymerase. We can talk about a little bit more of what tech polymerase is. DNTP, DNTPS or DNTP, DNTPs, buffer solution, magnesium, and potassium ions. So see, so we can put all these components into finally to our steps, how steps of PCR works. So these 3 steps are very significant, are very important for PCR. and this is basically all the steps we need, you know, and it's just repeat. It happens over and over and over to make, well, basically thousands and millions of copies of DNA. So first step would be denaturation. What it means is to heat. So you have to heat the copies of DNA, or a single or couple copies of DNA, to break up the double strand. Remember, the DNA coils up in a double strand, and they're actually pretty strong because they're hydrogen bonded. So we have to break those hydrogen bond with the mild heat, and those once it's broken up, you can go into annealing step. So the annealing primer. So we talked about primer to the single-stranded DNA template. So once that's done, elongation step comes in, and DNA polymerase. And we talked about tag polymerase here. And so usually it's tag polymerase that uses in PCR. I know I know there's cases that PCR they can be using in different DNA polymerase, but most of the time it's gonna be tag polymerase because it's very good polymerase for us to use in PCR. So it's very common. So to synthesize new DNA complementary to the DNA template shared by adding D NTPs that are complementary to the template in 5' to 3' direction. Like you guys learning in DNA replication and transcription translation, the direction is important because that's how DNAs are copied. So I hopefully you guys can think about the direction of how these are copied. You guys can actually go back all the way here. You know, you see this is copied 5 to 3, 5 to 3, always 5 to 3. So hopefully this video helped you guys, and like always, if you guys have any additional questions, please feel free to contact me, and I'll see you guys next time.